Hello, we're glad you've joined us for this live webinar, Chicago and Dovetail Hi-C Yield Chromosome Length Scaffolds of the Ixodes scapularis genome. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Dovetail Genomics. Dovetail Genomics is transforming genomics by making long-range genomic information readily accessible to all. The company enables researchers and clinicians to solve complex problems involving de novo assembly, structural variation, cancer research, and more by providing a comprehensive view of the genome. For more information on our sponsor, please visit dovetailgenomics.com. Let's get started. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. I now present today's speakers, Monika Gulia-Nuss, PhD, Assistant Professor in the Department of Biochemistry and Molecular Biology at the University of Nevada, Reno, and Mark Daly. Mark Daly is the Director of Business Development and has been with Dovetail for 2 years. Dr. Gulia-Nuss's research focuses on understanding the basic biology of ticks to identify novel targets for tick control. Her long-term goal is to develop novel strategies to control ticks and tick-borne disease transmission. To this end, she's generating first-ever transgenic ticks using the CRISPR-Cas gene editing system and employing other cutting-edge genomic techniques. These include Hi-C-based genome scaffolding for better assembly and annotation of the tick genome. Her work focuses on understanding the role of insulin signaling in tick-pathogen interactions and differences in the chemoreception circuit in different Ixodes species. Her complete bio is on the LabRoots website. Dr. Gulia-Nuss will now begin her presentation.
Great. So thanks a lot for the introduction. My name is Mark Daly, and I'm the Director of Business Development here at Dovetail. I've been with the company for about 2.5 years, and I'm very privileged to be a part of Monika's presentation today. I think it's going to be really exciting. Before Monika gets started on her slides, though, I just want to give everybody a brief 10 or 15-minute overview on the Dovetail technology, just so it kind of sets the stage for Monika. My email is here on the first slide, so if you do want to reach out to me with any questions, I also have a much longer version of my slides that I'm happy to present one-on-one with anybody in the audience. Just send me an email. I'd be happy to do that. Dovetail, all we do is de novo assembly. We've been doing it now for over 5 years, and we're specialists in de novo assembly. And we've now done over 1,000 de novos, most of those for actual customers. We've done quite a few internal projects as well. So we've really got a lot of experience in doing this. And what I'm going to do is take you through the process of building a de novo assembly at Dovetail. This first slide serves kind of as a primer for de novo assembly. I'm sure there's people on the line here that are new to de novo, and I want to make sure that everybody understands this information. So I'm going to explain some of the terminology as I go along. This slide also summarizes what Dovetail does in a single year. So we, in order to build a good quality, highly accurate, highly contiguous de novo assembly, we need to understand that we need to capture the genome on different scales. And on the left side of this slide, I've listed the 3 scales of genomic content: short range, mid range, and long range. And we go through this process in a stepwise fashion to build greater and greater contiguity until we get to a point where each chromosome is represented with a single scaffold. And a common terminology that you'll see in this presentation is the scaffold N50. And just for folks that are new to de novo assembly, the scaffold N50 is really the standard way of measuring quality of a de novo. It really represents, in simplistic terms, the median scaffold size of the assembly. So the lower the higher the N50 number, the better. So what I've shown here is this progression of capturing greater and greater contiguity. The initial short-range data is typically what would be represented in a draft assembly. So we can either take a draft assembly from you and improve it, or we can build one entirely from scratch here at Dovetail. But in either case, the draft assembly is typically built using a variety of methods, one or a combination of Illumina short-read technology, shown on the left, PacBio long-range technology or long-read technology, shown in orange there. 10x Genomics is a newer technology that we're starting to use at Dovetail that is a synthetic long-read technology. And there's also Oxford Nanopore, which is a newer long-read technology as well. And we will choose the appropriate technology based on our knowledge of the organism that you're working with. So, but it's really about selecting the right tool for the job. And in any case, however we build that initial draft assembly, we'll get it to a point where we can then apply a mid-range data type, which in our case is called Chicago. And Chicago reads are shown in red there. And Chicago reads are kind of like really big mate pair reads. They can span genomic content from between around a kilobase up to about 150 kilobases. So we're starting to capture a much greater scale of genomic content. And as you can see in my graphic here, we're starting to make joins between contigs that are quite distant from each other. So now we're starting to build scaffolds, which are shown in purple here, but we're still far from chromosome scale. So we need to employ a third technology to capture the long-range data, which in our case is called Dovetail Hi-C, and those reads are shown in black there. And Dovetail Hi-C is a very unique technology in that we can create megabase-sized reads. So we can literally span one telomere to the other and ultimately get you up to chromosome scale assembly. And that's really the ultimate goal of someone that is building a de novo. You want to really get to a point where each chromosome is represented with a single scaffold, and that's what we're able to do at Dovetail. We often use this final step called gap filling, which is taking low coverage PacBio reads and essentially filling in gaps where we're able to after the Hi-C step. So at the end of this whole process, you'll get an assembly that is not only highly contiguous, but also very accurate. We employ a variety of different de novo techniques to get that initial draft assembly, and I've listed them here. The most common ones are Illumina short-read technology, which is the least expensive, and it's also very widespread. But the caveat there is short-read technology is really only good for easy genomes, quote unquote, and easy genomes are typically birds, mammals, and haploids. If you're not working with one of those, then you really need to bring in some kind of long-read technology, which could be 10x Genomics, PacBio, Oxford Nanopore are the most common ones. And I've listed the advantages and disadvantages of each of those. And again, if we're building this assembly at Dovetail, we'll make sure we consult with you on choosing the best approach for your budget and for the level of complexity of the assembly that you have. Okay, so now we've built this initial draft assembly, so now we can start scaffolding it to full chromosomes. And this is where we start using the proprietary Dovetail technology. And we have three major types of libraries that we work with at Dovetail for scaffolding. They're listed here in this cartoon: Chicago, Hi-C, and Hi-C, Dovetail Hi-C. Now, Hi-C is a very specific technology that's really only used for formalin-fixed paraffin-embedded human tumor samples. So I'm not going to talk about that here today. If anyone on the call is interested in hearing about that, send me an email. For now, I'm going to focus on Chicago and Dovetail Hi-C. So these two technologies share basically the same molecular biology steps. Chicago starts with purified high molecular weight DNA, whereas Hi-C starts with tissue. So I'll start with Chicago. What we do with Chicago is we extract ultra-high molecular weight DNA, typically around 150 kb long. And what we do, and so what I've shown here in the cartoon, you can think of this as a 150 kb molecule here, and I'm following 2 points on this molecule labeled 1 and 2 through this whole process, so you can see what happens. The first thing we do in Chicago is we strip off all the protein from the DNA. And so we're removing all the chromatin. And then what we do is we build artificial chromatin in vitro. And the blue circles represent artificial nucleosomes. And there's a very important reason why we do this first step, which is that natural chromatin, for example, like you would use in Hi-C, likes to form looping structures. And those looping structures, while they may be of interest biologically, they will actually create proximity ligation events that are due to biology and not due strictly to linear distance along the molecule. And those biological events will create a lot of background noise that makes scaffolding much more difficult. So the beauty about Chicago data is that it's very clean data, and I'm going to show you that in a few minutes. I'll show you an example of that. So we've created this artificial chromatin. We then add formalin, which does two things. It condenses that molecule down into a globular molecule, bringing distant parts of the molecule close together. So you can now see 1 and 2 are very close together where they used to be very far apart. It also creates covalent crosslinks between the proteins shown in red there and fixes that molecule in space. In step B, what we do is we chop up that molecule with an endonuclease, creating sticky ends. In step C, we blunt fill those sticky ends and add a biotin label, which is just used for a downstream purification step. And then in step D, that's what I like to call the magic step, because we actually add a ligase, which then randomly ligates those blunt ends together. And essentially, you're going to get every possible representation of a ligation event for each of these molecules. In this case, you can see that 1 and 2 that used to be very far apart are now literally joined together. And that's going to be very valuable long-range proximity data that we're going to be able to utilize. In step E, we release those crosslinks, and now we have all of these proximity events floating around in solution. You can see that in our example here, 2 of these proximity events are of real interest to us, the top and bottom, because those 2 represent internal ligation events. The middle one is not of interest because no internal event occurred. So we'll just ignore that middle one. But we're going to then take these internally ligated events and sequence them with traditional Illumina PE150, paired N150 sequencing. And we're going to take all of those proximity sequence events and put them into our HiRISE software. And HiRISE will take your draft assembly and the Chicago data, and it'll attempt to make long-range connections utilizing that proximity data. And so now we're starting to build contiguity in this assembly. Dovetail Hi-C does a very similar, the process is very similar, except we're starting with tissue instead of DNA. And so you can imagine that in step A there, instead of this being a single molecule, this is actually an entire chromosome. And so this process of proximity ligation occurs throughout the entire chromosome from one end to the other. And thus, when we take that data and put it into HiRISE, we're now going to start building contiguity up to a full chromosome. So let's now take a look at the data that comes off these two different libraries quickly. So this first slide showing the data shows on the x-axis the genomic coordinates. Notice the two scales are different. This first chart goes out to 0.5 megabase. The right-hand chart goes out to 100 megabases. The vertical axis is the same in both charts. It's the proportion of proximity events of a certain size. So the Chicago data is charted in green, and if we go to the very top of the Chicago curve, these are proximity events that are a few kilobases apart. As we go down this curve, the events get farther and farther apart until we get to the very bottom here, and this represents the longest molecule in the sample. So these are proximity events that are around 150 kilobases apart. So in Chicago data, we have this very deep coverage of the genome between 1 kb and 150 kb. The Dovetail Hi-C chart or curve is totally different. It's much flatter and goes out well beyond 0.5 megabase. In fact, we have to zoom out to the right-hand chart to see that Dovetail Hi-C data actually goes out to the end of the chromosome. So you get this incredible coverage covering the entire chromosome and very even coverage, and these curves are very reproducible from organism to organism. You can see that on the right chart, the scale is so great that the Chicago data is barely visible. So one might ask then, well, if Hi-C data is giving me full chromosome scale contiguity or coverage, why do I need Chicago data? What's the point of having Chicago data if Hi-C is giving me full chromosomes? And there's two very important benefits of Chicago data that I'm now going to talk about. The first big benefit of Chicago data is that it boosts your input assembly contiguity to a point where Hi-C will effectively work. It turns out that in all our experience of doing this hundreds and hundreds of times, that Hi-C really requires an input N50 close to a megabase to work effectively. And in this example, which is a weeping love grass, which is a 600 megabase genome, you can see that on the top table, the input assembly had an N50 of 380 kb, 0.38 megabases. When we applied Hi-C only to that assembly, we only improved the contiguity to about 5 megabases. This is far short of chromosome scale. So this was really a failure, right? We did not assemble contiguity level, chromosome level contiguity in this assembly. What we did then is we did Chicago first. So we took that input assembly and applied Chicago data to it, boosting the contiguity to closer to a megabase, 0.791 megabases. And then we took that assembly and ran it through our Hi-C process. That's when we achieved chromosome scale contiguity, as shown in the very bottom table. We got a contiguity of 45 megabases. That was chromosome scale. So Chicago data is needed as a mid-range step in between your input assembly and the Hi-C assembly. The second major advantage of Chicago is in accuracy. So now I'm going to shift gears from talking about contiguity to talking about accuracy, because there's no point in building a very highly contiguous assembly if it's full of misjoins and errors. What I'm showing here now is a comparison of the same region of a genome using Chicago data on the left and Hi-C data on the right. The vertical axis is the size of the proximity event going from 1 kb at the bottom up to 200 kb at the top. The very first thing you notice is that Chicago data has far higher density than Hi-C data. Each purple dot that I've shown there is a proximity event. This makes sense because with Hi-C, we're trying to scaffold entire chromosomes, whereas with Chicago, we're only having to assemble molecules of 100 or 150 kb. So this data density difference makes a lot of sense. So what we then did is we introduced a false misjoin into the input assembly and then tried to scaffold that with Chicago and Hi-C. And the misjoin is actually an inversion event that we purposely created, and it's shown in red here. And when HiRISE sees this data, it's not able to properly position the Chicago reads because of the false misjoin. So those reads get displaced, and the data gets displaced in this bow tie pattern that I circled here. These signatures, they stand out like a sore thumb. They're very diagnostic of a false misjoin or a misjoin. You notice that in the Hi-C data, in the same region of the genome, there's no difference between the top and bottom plots. In other words, Hi-C data is not able to to allow HiRISE to find these misjoins. The reason why is that it doesn't have a high enough density. So what Chicago is providing you with your assembly is not only boosting contiguity, but it's also allowing HiRISE to identify and correct false misjoins in the input assembly, thus giving you a far more accurate final assembly than if you did not use Chicago data. The next slide shows you what the raw data looks like, comparing Chicago on the right to Hi-C on the left. You can see that the Hi-C data is very noisy. Those squares that I've shown with arrows represent TAD domains. Those are the looping structures that form in natural chromatin. Again, very interesting to a biologist, but they really confound de novo assembly. The data on the right is extremely clean, and that is why Chicago data is so powerful in allowing us to properly order and orient contigs in your input assembly and correct misjoin events. I'm almost done with my slides here, just a couple more. This slide shows you the progression of de novo assembly going from the input de novo on the left, where we have tens of thousands of scaffolds that lie along the diagonal here. The midpoint, which is Chicago, we're now starting to build megabase size scaffolds shown in dark blue, those squares along the diagonal. But we still have lots and lots of smaller scaffolds, so we're not nearly at chromosome scale, but we're building up the contiguity. Then on the far right, when we add in the Dovetail Hi-C data, we finally get single scaffolds for each chromosome represented. We've done, like I said, hundreds and hundreds of assemblies of all kinds of taxa, mammals, reptiles, plants, fish, insects. We've done polyploids, highly heterozygous assemblies. And this slide kind of just shows you a brief sort of cross-section of some of the genomes we've done. And I'm showing this to just basically demonstrate that regardless of genome size, regardless of the input N50 that we start with, we virtually always end up with chromosome or near chromosome contiguity. And you can see the levels of improvement here are pretty staggering in some of these, up to 7,500 X improvement in contiguity. I just want to end my presentation with just a note on our new Matching Funds Award program. We're building this tree of life, which actually should go up on our website pretty soon. We're up to around 350 organisms, but we would love to increase the size of our tree of life, with your genomes. And so what we're doing here is we're offering this Matching Funds Award. You can apply at the website here, or send me an email, and I'll make sure that your application gets entered. And the purpose of this is really to help offset the cost of de novo assembly. We'd love to get to a point where every organism on the planet has been sequenced, and we'd love to be a part of that. So if you're interested in applying to this award program, just let me know. So with that, I appreciate your listening to my slides and my presentation. With that, I will pass it over to Monika, and she will present her tick assembly project that she did with us.
Thank you, Mark, for a very informative overview of the technique. I would also take this opportunity to thank Dovetail Genomics for funding this project and doing an incredible job at it. This project is a part of the bigger project that we have been working on, sequencing tick genome. So I'm a vector biologist. I'm an assistant professor at the Department of Biochemistry and Molecular Biology at University of Nevada in Reno. My lab is interested in understanding arthropod biology in general. We are interested in arthropod pathogen interactions and identifying novel targets for disease vector controls. One of the major areas of research in my lab is to develop new molecular tools for tick research. And I will talk about this genome assembly here today. So I was very fortunate to be part of the Tick Genome Consortium and was the first author on the published tick genome paper. This tick genome that we published in 2016 was a decade worth of work, and the sequencing took place pre-next-gen sequencing era. So that was really not an easy feat because it's a complex genome, and I will talk about that during my presentation here. So I have this presentation today. I will talk about tick life cycle first, and then I'll talk about the importance of studying tick vectors. And then I will give you a little background of tick genome. The first assembly is Ixodes, or this is I. scapularis Weichel strain 1 assembly. Then I will talk about need for a new assembly, and Chicago and Dovetail Hi-C assembly that we just did with Dovetail Genomics, and what the next steps are in this process, and then I will be happy to take any questions later. So our main objective for doing this project or carrying out this project was to obtain a chromosome-level scaffold assembly for a highly repetitive genome. We really wanted to improve the annotation using a highly contiguous genome, and as Mark mentioned, that with the Hi-C you can get highly contiguous genome, and this is what probably everybody needs for their genome. We wanted to improve gene synteny analysis because right now our genome is quite fragmented, and I will talk about that in a bit. So first I would like to talk about the ticks, our model organism. The tick that we work on is black-legged tick, or Ixodes scapularis. It's a very important vector of Lyme disease. Ticks have 1 to 2 year of life cycle, or especially I should say the black-legged tick has 1 to 2 year of life cycle, depending on where they are, the geographical area. In northeast, where it's colder, life cycle is usually 2 years. In southeast and in Pacific West, it's usually a year of life cycle. Each life stage of tick requires a vertebrate blood meal. So that means when the ticks hatch from eggs into larvae, larvae require a blood meal, and then they molt into nymphs, and nymphs require a blood meal to further molt into adults. Then males of this species do not feed, whereas the female feed to reproduce. So here you can see that there are multiple time points in their life cycle when they can acquire a pathogen and transmit this pathogen to the next organism when they are feeding on that organism. Most infections, most bacterial infections in particular, are interstitial. That means they carry over from one stage to another stage after molting. For example, if a tick larva get infected, the resulting nymph will be infected as well. So why are we studying tick vectors? Well, as I said, tick vectors are quite important. Hard ticks or Ixodes group, these are vectors for rickettsial, spirochetal, viral, bacterial, protozoan diseases. Tick causes or tick carries the pathogens for more diseases than any other organism. They can also, not only the pathogens, but they can also lead to severe toxic conditions, for example, paralysis and toxicosis. Lyme disease itself infects more than 300,000 individuals every year in the United States. And Lyme disease is the fastest growing vector-borne infectious disease in the United States. So there's a big need for understanding these vectors. And when we are trying to understand these vectors, one of the areas that we are lacking is the availability of a good genome. So this is the first assembly, the ISCA-W1 assembly that was published in 2016. And this is the only tick species that has been sequenced to date, so Ixodes scapularis. The problem with tick genomes is they're quite large. For example, Ixodes scapularis genome is 2.1 gigabase pairs. And another complexity that they add is there's a highly repetitive genome. The sequencing that we carried out, the sequence that was published earlier in 2016 was done with backs and backend sequencing, which resulted in 3.8-fold coverage. But it was a highly fragmented genome, as I mentioned. It was fragmented into 370,000 scaffolds approximately, and N50 of a scaffold was approximately 50 kilobase pair. There were 20,000 protein coding genes were identified in the genome. Scapularis preliminary fish-based karyotype resulted or predicted chromosome number 2n equals 28. So there are 28 chromosomes. They have X and Y chromosomes as well. So this ISCA-1 assembly was done with embryos of Weichel strain, and that's where the W comes from. So when as I said, that this assembly was highly fragmented with 370,000 scaffolds. This highly fragmented assembly makes it really difficult to do any transcriptome assemblies. So most of the transcriptome assemblies that have been done on ticks, on any of the Ixodes species, have been de novo assemblies because the existing genome is it's too fragmented to confidently align these transcripts to the genome. This also makes it it also complicates the ortholog identification because the genomes are quite fragmented and our gene models are incomplete. The good gene synteny N50 needs to be higher, approximately 1 MB or higher, as good for gene synteny analysis. So a highly contiguous genome was needed for correct gene annotation and synteny analysis. So that's where we contacted Chicago. We contacted Dovetail Genomics to carry out Chicago and Dovetail Hi-C assembly, and we were fortunate to get this project funded through matching grants. So this is, I'm not going to go over the details of Chicago and Dovetail Hi-C because Mark already gave a very good overview of these, how these techniques work. So what we got from Dovetail Genomics was, so when they sent us the results, these results were incredible. Here you can see that L50 and 50 of Dovetail Hi-Ride assembly resulted in 12 scaffolds that were 48 MB. These out of, and there were several, a lot of scaffolds were over 1 MB. So these are their biggest, their largest scaffolds here listed. L90 and 90 resulted in 2,895 scaffolds of 4.030 MB. So here, if we look at this graph, this is a comparison of the contiguity of input assembly and high-rise scaffolds. Each curve here shows the fraction of the total length of assembly that's present in a scaffold of a given length or smaller. The fraction of the assembly is indicated on the y-axis here, and the scaffold length in base pair is on the x-axis. These two dashed lines here represent N50 and N90 length each. So any scaffold that was less than 1 kb was excluded from this analysis. So these are all over 1 kb scaffolds. Here, if we look at this link density histogram, you'll see that here all the scaffolds that were less than 1 MB were excluded from the analysis. And the x-axis and y-axis are mapping positions of the first and second read in the read pair respectively. These are grouped into bins. The color of each square gives the number of reads pair within that bin. White vertical line and black horizontal lines have been added to show the borders of the scaffolds. And as I said, all the scaffolds less than 1 MB were deleted from or were taken out from this analysis. So this link, when we looked at these scaffolds, these long scaffolds, there were 28 long scaffolds. Those were over 1 MB. And this number 28 corresponds to the chromosome number, so which suggests that we have chromosome level assembly of tick genome here. So here, if we compare this assembly statistics with the original ISCA assembly, this is amazing here to see that we went from approximately 370,000 scaffolds that were over 1 kb to 82,672 scaffolds with high-rise assembly. The Contig N50 was 2.94 kb in our original assembly, which improved to 64.21 kb in high-rise assembly. And then we have the percent gaps in the percent genome in the gap was approximately 49% with our ISCA assembly. So we have only 51% of genome was sequenced. Here with the high-rise assembly, we have approximately 3% of genome that's in gaps. So this is a remarkable feat to achieve, and this now gives us hope to improve our annotation well. So the key takeaway from this presentation or this project is that Dovetail Hi-Rise assembly can give you very high very long scaffolds. So our analysis or our data resulted in 12 scaffolds of over 48 MB. There were 20 scaffolds that were over 1 MB, and this number corresponds to the number of chromosomes in tick genome. The genome synteny, as I said, requires a scaffolds of over 1 MBP, and we are in a good shape now to do the synteny analysis, and hopefully we will have a very good annotated genome. So successful contiguous chromosome level assembly of a highly replicated genome was possible because of this technique. So the major key takeaway from here is that because we have now this ability to rapidly and reliably generate genome assemblies with chromosome length scaffolds for non-model organisms, this would accelerate genome analysis of many organisms, and that includes other tick species as well, not only Ixodes, but we might be able to now get chromosome length scaffolds and assemblies for even bigger tick genomes, such as Syphilis micropolis, that genome is approximately 7 GBP. So this gives us a lot of hope. So what's the next step for us? We would like to fill the gaps now either with 10x Genomics or PacBio. Since we have very few gaps, our genome is about 3% of genome is in gaps. So probably 10x Genomics will be an inexpensive option for us to fill those gaps and then reassemble the genome, hopefully with high-rise scaffolds with Dovetail Genomics again. We would, once we have this contiguous genome, we would like to map our existing gene models to the new assembly, and hopefully we will have full-length genes available. And with this, I am happy to take any questions.
Thank you, Dr. Gulia-Nuss, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Gulia-Nuss will answer as many questions as time permits. The first question is, how should samples be prepared before sending to Dovetail?
Okay, it's Mark Daly here. I can take that question. So basically, the most important thing is that samples should be flash frozen on liquid nitrogen and then shipped on dry ice. We do have a sample guidelines document for both Chicago and Dovetail Hi-C that I can send anyone on the call that's interested in getting that. Just send me an email and I'll send that to you. But the most important thing is being flash frozen. We have tested a variety of other sample prep methods like ethanol and RNA later and lyophilized tissue, and they are kind of hit and miss. They don't always necessarily get high quality DNA for Chicago. So just use caution with those other kinds of sample prep methods.
Thank you.
Next one is, oh, I'm sorry, Monika, please.
No, I was just going to add to that. For our samples, and I agree with Mark, flash frozen samples work great for most anything that we do in our lab as well. For our samples, we sent we sent 6 sample sets, 3 replicates of each, because we didn't know whether they will be able to get good DNA amounts from adult ticks or eggs. So we sent both egg clutches as well as ticks, and the embryos were used for these experiments. We are quite fortunate that ticks lay a lot of eggs, so they lay about 2,000 to 3,000 eggs each female. So we collected the whole clutch of a female. So there were 3 different females that laid full clutch, and we sent 3 different sets of samples, flash frozen in liquid nitrogen and sent on dry ice.
Yeah, that's a good idea. That's a really good idea, Monika, to send generally more sample than we necessarily need. We can always send it back. What we don't want to do is, like Monika said, we're in the middle. We do an extraction, and for some reason it didn't work, and then we have to go back to you to get more tissue, and that will delay the project. So our project managers will give you the specific guidelines based on the kind of organism you're working with.
Thank you. Thank you both. Next one is, what if my scaffold N50 is less than the recommended minimum of 20 kb?
Okay, so in that case, so we have this minimum requirement for an assembly to go into Chicago, that it has to be a fairly good quality assembly. It can't be too fragmented. And our minimum threshold for that is a scaffold N50 of 20 kb or higher. Most of the projects that we do where they're improving an existing de novo assembly that our client sends us, most of our projects, the N50s are higher than 20, but we have occasionally got some that are lower. There's 2 options there. Either you can risk running the scaffolding on your more fragmented assembly. We do have examples, and I think there was a slide in this presentation of some projects where the N50 was lower than 20. I think we did one of the turtle project where it was, I think, 17 kb, and it worked really, really well. It's just that it's not as likely to perform than if you had a higher N50. So the second option is that we would recommend improving that input N50 by adding more sequence data to the draft assembly. So for example, if you used strictly Illumina sequencing for your input assembly, we might suggest using a longer insert and doing more short read sequencing, or even better, adding some long read sequencing like PacBio or 10x Genomics or Oxford Nanopore as an example. to get that initial N50 above the 20 kb threshold, at which time then we can go into this Chicago pipeline. One other quick thing is that Hi-C really requires an input N50 of a megabase or higher. So the 20 kb metric is strictly for Chicago. If you're attempting to do Hi-C on an input assembly with a scaffold N50 lower than a megabase, it's not nearly as likely to get you full chromosomes than if your input N50 was a megabase or higher.
Great, thank you. Next one is, what challenges do you face when doing a new project?
I think,
would you like to take that question?
Yeah, okay, sure. So like I think Monika addressed one of the challenges already, which is, with something as small as a tick, how do you minimize heterozygosity? And, for these, for doing a scaffolding project, we need at least 500 nanograms to a microgram of DNA for the Chicago step. And we also need, obviously, tissue for the Hi-C step. And, Monika was saying that luckily a single tick produces, so many eggs that we don't have too much of an issue with that with her project. But we have, there's basically 4 challenges at the draft de novo stage. So when we're building an assembly for our clients, the main challenges are heterozygosity, genome size, repeat content, and ploidy. And those are things often we can't control those, right? I mean, the only thing we can really potentially minimize is heterozygosity. And we have various ways to recommendations regarding that. Another kind of challenge is just sample quality. Getting really good DNA, high molecular weight DNA for Chicago is really critical to the final success of scaffolding. So if ideally we want DNA that's around 150 kilobases long for Chicago, and if we're only able to extract DNA that's 75 kilobases, Chicago won't scaffold as well. A final kind of main challenge that we face is just if a client is sending us an assembly to improve, we don't really have any control over the quality of that assembly. We will definitely QC it and make sure that we believe it's sufficient quality to scaffold. But having a good quality starting assembly is also really important for final success.
Thank you. Next one is, how is Hi-Rise better than other scaffolding softwares?
Okay, that's a great question. So Hi-Rise is a proprietary software platform that was developed by Nick Putnam at Dovetail, and the input data types for Hi-Rise is either Chicago or Dovetail Hi-C data. There aren't that many scaffolding softwares out there. There's lots of de novo assemblers. In terms of, we have done a direct comparison with Salsa2, which is a publicly available scaffolding software tool. And we found when we did a very controlled experiment comparing Hi-Rise with Salsa2 that Hi-Rise was more accurate. It's more conservative. We got fewer errors. Salsa2 admittedly seemed to provide a little bit higher contiguity. But Hi-Rise was more accurate at the end. And I have that data. If anyone is interested in seeing that experiment, I can provide that. The other thing I want to emphasize too is that Dovetail is the only provider of the Chicago service, and that is a very unique data type, as we talked about in the introductory slides, in that Hi-Rise uses Chicago data to find and correct misjoins in the input assembly. And that is a unique feature of Hi-Rise that makes the platform very, very powerful.
Thank you. The next question is, does our draft assembly need to be built in any particular way before handing off to Dovetail for scaffolding?
No, if someone says of the draft assembly to scaffold, we're agnostic as to how that assembly is built, as long as the minimum threshold is met of 20 kb for Chicago and 1 megabase N50 for Dovetail Hi-C. But whether you use, it doesn't matter what kind of sequencing platform you use, and it also doesn't really matter what kind of assembler you use. What we like to have, though, is the most accurate draft assembly that you can provide us. And the more accurate and less fragmented the input assembly is, the better Chicago and Dovetail Hi-C will just work that much more effectively.
Thank you. The next question is, if I could only do one, which is better, Chicago or Dovetail Hi-C?
Okay, I can take that question. So it really depends on starting contiguity. So if you only could do one of those two methods, if your N50 of your input assembly is less than a megabase, for example, 100 kb or 200 kb, then I would highly recommend Chicago. Because as I said, Hi-C ideally needs an N50 of a megabase to work effectively. If you have an input assembly where the N50 is very high, like a megabase or greater, you'll probably get more bang for your buck doing Dovetail Hi-C.
Okay, thank you. Can I sequence your Hi-C libraries myself?
Yes, you can, definitely. These libraries are standard Illumina libraries, and so you can sequence them on any of the Illumina Hi-Seq platforms. We use the Hi-Seq X10 when we do it for you at Dovetail as a service, but certainly we can ship the libraries to you, and you can sequence them, and then you would upload the raw data to Dovetail for the next step. We would provide you recommendations on how many read pairs to, what coverage depth to use on a particular library. So we can consult with you on that so that you ensure that you get enough sequence coverage.
Great. Next is, now that you have this tick assembly, what are the next steps for your research?
That's a great question. So as Mark earlier said, I'm going to start from the previous question, just add a little bit on the challenges in getting a good assembly. So we work on non-model organisms, and non-model organisms, as you all might know, don't follow the same rules as the model organisms most of the time, and that's quite a big challenge. So when we started working on this genome project a decade ago, we ended up having this genome, at least a genome of the first tick, any of the tick species so far. This is the only genome. We had a very fragmented genome, but now with Hi-C, thanks to Dovetail, with this new data, that we have a much better genome assembly. This gives us we are very hopeful that we will be able to improve it even further, and we already have a chromosome-level assembly. Now the next challenge for us is to annotate this assembly or bring our annotation to this assembly. That will help us correct our gene models, because most of our gene models right now with our existing assembly are incorrect or incomplete. So this will allow us to complete these gene models, correct the gene models, and also we will be able to now bring our transcriptome assemblies to this genome assembly. Another really good feature of a chromosome-level assembly is we will be able to now make more physical mapping per se. I'm using it a little loosely, but we will be able to see how the genes are arranged on the chromosomes and which genes make a group. So the synteny will be improved quite a bit, that I'm hoping. So yeah, for next step for us is to annotate this new gene assembly with our existing annotations and correct our gene models and complete our gene models, make it a really good assembly that can be used by anybody to do their transcriptome assembly, do single cell assemblies. All these different areas are now open for tick research. In my lab, we are focusing on editing ticks, so we need correct gene models in order to make our guide RNAs for CRISPR. So this will also allow us to find the correct genes or the complete gene models to be able to do the next steps. So really, this is going to open a lot of doors, not just for our group, but for the tick community in general. We are already getting a lot of requests to publish this assembly so people can use it, and we are in the process with Vector-Base. We will be publishing it on Vector-Base, so it's open access. Everybody can use it. So yeah, really, I think in short, I can say that this is going to push the tick research in terms of genetics and genomics forward, and we are hoping to see more and more tick species sequenced and assembled to chromosome levels.
Thank you. Looks like this may be the last question we have time for. What coverage of PacBio do you suggest?
Yeah, I'll take that one. So PacBio is one of the options for building the initial draft assembly. And if someone is looking at using PacBio, we really recommend around 60x coverage to get optimal results. We have done a bit lower. 50x coverage is probably acceptable, but anything below that, we'd probably not want to attempt.
Thank you. I would like to once again thank Dr. Monika Gulia-Nuss for her presentation. Do you have any final comments?
Gosh, final comments. I think I would just like to say, if you have a fragmented assembly which is good enough to be able to do chromosome level assembly, you might want to just give it a try, because this will change the landscape completely once you have the chromosome level assembly. And we are very fortunate to get this done, because we weren't really hopeful that our assembly would work, but we are very pleased with the results. So,
thank you. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Dovetail Genomics, for underwriting today's educational webcast. This webcast can be viewed on demand through April 2019. Leverage will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Okay.